Let us begin our study of the microscope. We're going to start off with these guys here, the ocular lenses. Ocular means eyes, and this is where you're going to place your eyes. This is a binocular scope. You need to use both eyes. Do not use one eye, use both eyes. One of the things that you can do is separate and manipulate these slightly. They're very loose and easy to manipulate so that if your eyes are spaced more apart or closer together, you can work that out. The ocular lenses add a 10x magnification to everything that you look at, 10 times the magnification. We're now going to take a look at the objective lenses. Students are real tempted to move from one objective to the next by grabbing them like an udder and moving them around. Please do not do that. As you can see, I use a grip to move them that is up above, and it's actually very satisfying. You can kind of roll it forward and then it clicks into place. The red objective is the 4x magnification, 4 times the magnification. This is also known as the scanning objective lens. The yellow objective is 10x or 10 times the magnification. This is known as the low-powered objective lens. And the blue would be 40x or 40 times the magnification. This is known as the high-powered objective lens. Now we turn our attention to the stage. Your sample on a slide will be placed perfectly within this little metal bracket. I have seen all kinds of ways that students place a slide inside of here. I will show you the proper placement, but just so you know, there's this little arm on the side here, which usually has a lever attached to it, but it simply snugly keeps your slide within this metal bracket so that it perfectly fits within there. Please do not try to take this and strap it over the slide, for example, or somehow rest the slide anywhere other than where it's supposed to be, where it perfectly fits within this little structure. These two little knobs on the side here allow you to move your stage front to back, back to front, side to side. So you never actually have to touch your sample after it's on the stage. You are able to move the entire stage in the way that you want to. It's called an XY manipulator or a mechanical stage manipulator. Now comes the moment we've been waiting for. We're going to plug in our scope, click it on, turn the light up all the way. You can adjust it using the diaphragm later, which I will show you. This little lever down here is a diaphragm that allows us to control the amount of light coming up through the stage from our light source. As you can see, brighter or darker. Our sample today is going to be human blood smear, which was preserved ahead of time, a fixed slide. We're going to take our slide and place it firmly and snugly within our arm. Notice it fits perfectly. Now there are two focus knobs on the side. The one we're going to be using first is called the coarse focus knob, only to be used when you have the red objective or the 4x objective over the stage, because the 4x objective is short enough that the other objectives are longer, and it's possible that if you move the stage up and down too quickly, you could actually damage those objectives. There's no chance of that happening with the 4x objective because it's quite short. So you can move this. It'll move your stage quite quickly up and down, and that is actually appropriate at this stage. Once your stage is up fairly far, close to your 4x or red objective, is you'll be moving and manipulating the stage so that the sample, the light is showing directly through the sample. In this case, this sample is pink, so you'll be looking for kind of pink light to be shining up through the sample. And you're going to be eyeballing this from the outside. Now I'm doing my best to show you what you'll be seeing through the scope. I have a camera up, so it's a little bit shaky. My camera is showing that as you bring it into focus, you're going to be seeing some specific dots here in this case. It's not always easy to know exactly what you're looking for, but you should be able to see something that if you move your XY manipulators, you'll be able to see it moving. Otherwise, you might be focusing on sort of an artifact of the slide. In this case, you'll be looking for pink and purple dots that are fairly clear. We're going to now move to the 10x objective, and we're going to change our focus. Do we use our coarse focus? We do not. We do not use the coarse focus. We only use the fine focus at this point, the knob in the center. These microscopes are parfocal, which means that they are approximately the same level of clarity from one objective to the next. You may have to make minor adjustments, but never use the coarse focus unless, of course, you're on the 4x or scanning objective. Right now, remember that we are on the 10x or low power objective, and therefore it hangs quite far down. It's possible to damage that objective lens when you aggressively move the slide up using the coarse focus. And here we go. We're going to use just the fine focus and we'll be able to pull it into more clarity. Sorry about the shaky camera again. I did not have a teaching scope, just my own little camera. And there are these beautiful cells that we're taking a look at. Now we're going to switch to our 40x blue high-powered magnification, high-powered objective lens. And again, when we're taking a look at our focus, we do not use the coarse focus. No, not the coarse focus, only the fine focus. And being able to just slightly move the focus back and forth, we're able to bring, oh my gosh, it's a beautiful neutrophil. I specifically used this particular ocular lens because one of the ocular lenses has a pointer in it, and you can see that little black pointer there, so you can look at exactly the cell that you want to look at. And I'm going to show you what it looks like when we manipulate the mechanical or the XY manipulator, and we move it back and forth, up and down. Most of the cells we're looking at here are red blood cells, but there's our beautiful neutrophil. So gorgeous. I'm also going to show you what it looks like when we play with the light a little bit. Depending on what kind of a sample you're looking at, when you open up the diaphragm or close down the diaphragm a little bit, you'll be able to adjust that light and maybe see a cell a little bit better. Sometimes, ironically, under less light, you'll be able to see better under less light conditions. We have now finished looking at our sample and sketched whatever we need to see, seen whatever we need to see, count the cells that we need to count, etc. There are a few steps that you must follow every time you put a scope away. These are worth like $1,000, so please be careful with them and please make sure you're putting it away correctly every time. You are going to make sure that your 4x objective is in place over the stage. You're going to lower the stage all the way, remove the sample, turn the light source down all the way, click it off, unplug. Oof. And then you're going to wrap the cord of the electrical cord up and the back. Oh, this is hard to do with one hand. And stick it in there. Satisfying. You're going to cover it with the cover. Pop the sample back into the box. Also difficult with one hand, but there it is. I can do it with one hand. You can certainly do it. And click it closed. Well done.